Hi, so this is Lili Yang from UCLA. Sorry, I cannot join you in person at Munich. Hope you all have a wonderful conference over there. But I do appreciate Rick and the organization committee inviting me, give me the opportunity to share with you my research through the recorded lecture. And later on, I'll join you at the panel discussion. Today, I'm going to talk about advances in creatine and cancer therapy. Here is my disclosure. From this conference, it's nothing new anymore, and you'll hear a lot about like creatine and how it's wonderful for health, for beefing up muscle and increase strength. And those are in public's eyes. And I would like to bring up quickly about what's creatine scientists' eyes. Here is the molecular structure of creatine, and here is a biosynthesis of creatine in our body. Two organs are involved, kidney and the liver, both are needed. And for the creatine molecular wide, the couple of molecules involved, in its biology and the function, right? And especially, creatine very useful for the high energy demanding cells. We heard about muscle and brain cell. That's another type of cell really needed. And my specialty is in the immune cell. Those are all the very actively working, cells. They need energy, and creatine is a good energy source for them. And the particular molecule my lab has been studying, like in the past few years, is called creatine transporter. It is a transporter stay on the surface of the cell membrane, and its sole responsibility is to bring creatine from outside of the cell inside of the cell. And once getting inside, creatine will convert between two versions, the creatine and phosphocreatine. And that's a enzyme bidirectionally, working on that called creatine kinase. It can either, put a high energy phosphate on creatine, to store energy as phosphocreatine, or it can go the other way. It can release ADP and, convert the phosphocreatine to creatine, and by means release a molecule of ATP, which we all know that is the bio, energy, currency, like for the cell to work. And so for the deficiency of a creatine transporter, they are exist in human population, and that's disease consequence, following that. And today, I'm mainly talk to you about my lab's research on how creatine, and starting from how the creatine transporter, control the immune cell function. In the past few years, my lab has been focusing on the T cells. T cell is a very important part of the immune system. In the fight against cancer, they form the major army called the CD8 cytotoxic T cells. So their function is critical, right? And our research in my lab find that creatine is very important to fuel the energy, of the T cells during the fight against tumor, okay? And here list some lab members, two talented postdocs, Stefano and Jesse, and a talented senior scientist, Dr. Bowley, and the three of them has contributed to this set of the work I'm going to share with you. The story starts by, we are quite interested in, looking into the metabolic control of the immune cells in their fight against the tumor, and the creatine transporter, that molecule pop up at one of the screening, and that is, leading to our follow-up study. And here's what I'm talking about. So what we did is that we use animal model like carry tumor. We isolate the tumor from that animal, and we look into the cellular component of that tumor, and we pay attention to the tumor infiltrating immune cells, right? And then we do the qPCR and RNA-seq screening to look what changes those cells has encountered after they see, tumor. And if we see a significant change, kind of indicate that that molecule, or gene encoding that molecule play a role in the immune cells fight against the tumor. So creatine transporter is one of the molecule really are kind of standing out. Here shows that this bar over here, so this are the level expressed in the tumor infiltrating immune cell compared to the cells outside of the tumor and compared to the same kind of cells in a tumor-free animal. So you can see a dramatic change. We immediately interested in thinking that, seems that creatine transporter may play a role, in those cell function, but how, right? So like, our immunologist, and biologists in general, we use some very classical tools help us to understand, the precise function of a given molecule or the gene encoding that molecule. And those two are called the knockout mice. Knockout mice work in a way, it's an intact mouse, but it's deficient of one single gene. In this case, we knock out the creatine transporter. And then we compared that animal with the animal is wild type, is competent, for this creatine transporter gene. And we challenge those two type of animal with tumor. And to our, surprise, we find a really big change. Okay, so in the deficiency of a creatine transporter, the tumor grow much faster. Okay, then we asked whether it's because of the immune cells do poor job in the situation. So we look into the tumor growing in each of the animal, and we isolate those immune cells, and we run some assay called flow cytometry. So look at the activation status of those T cells. And we indeed find in the absence of the creatine transporter, those cells upregulate exhaustion marker much more significantly than the wild type cells. So it suggests like a PD-1 molecule here suggests that they are more, a kind of a prone to become, become inactivated after they see the tumor. So that, is quite interesting, right? So it links creatine transporter directly to the anti-tumor T cell function. But is that the ultimate, evidence to show that link? No, because for the whole body creatine transporter knockout mice, every single cell is deficient of the creatine transporter, right? And the tumor can have many components. The tumor cells themselves is surrounded by fibroblasts, stroma cells, and many other cells. So we wonder that indeed, creatine transport matters for the T cells. So again, we employ another very smart strategy, very classically used by the immunologist to dissect this question. What we did is that we breed the knockout animal to a kind of T cell receptor transgenic animal. That T cell receptor transgenic animal have one specific type of T cell. We design it in a way those T cell recognize tumor antigen, okay? And then we isolate those special T cells from the animals, either breed into the creatine transporter knockout background or breed into the wild type background. And so we isolate T cells. Remember now, only T cells will be deficient for the creatine transporter. And then we transfer those T cells into a wild type animal carrying tumor. So in this entire setup, okay, in the tumor microenvironment, the only single cell that's deficient of creatine transporter are the anti-tumor CD8 T cells, okay? And then we look at, okay, now that everything is the same except for the T cells, whether creatine transporter matters for the anti-tumor response. Indeed, it's a case. So here we see that for the knockout, CD8 T cells, they cannot really control the tumor growth as well as the Y-type compartment, okay? And we look deeper into their phenotype and function using flow cytometry that indeed confirmed those cells, responds really poorly to the tumor, stimulation. So they proliferate less and they make less, effector molecules fighting off tumor, and they are easier, to become inactivated, after the tumor encounter. So then it comes to the next question. How, right? Creatine transporter, it's just a transporter. Its function, we know, it's transport creatine into the cells. So why does that matter, for CD8 T cell response? So we designed a series of the in vitro and the in vivo study dissect that function. At first, we isolate CD8 T cell from the Y-type versus the creatine transport knockout animal, and we culture those T cells in the tissue culture dish. And we looked at every aspect matters to a T cell function. So we find indeed creatine transport deficiency decreased proliferation of T cells, seems not to do too much for the survival of the T cells, but indeed, matters a lot for the T cell function. Here shows that the very important effector molecules like IL-2, interferon gamma, and those are greatly, reduced in the absence of the creatine transporter, okay? And as well as other surface markers like CD25 and other like cytotoxic killer molecule important for their function to kill tumor, called granzyme B over here. Okay, and we also did a quite nice, rescue experiment. So the logic behind this is now we have a creatine transport deficient T cells cannot do their antigen response, work very well. Is that because of the development or because of creatine transport directly matters for their function? So this can be answered by giving back the creatine transporter, gene to the knockout cells and see whether it can rescue their function. And indeed, when we give back the creatine transporter, we can rescue the function. So which means creatine transporter directly regulate the function of the mature T cells, okay? That's independent of the development regulation, okay? And now let's dig deeper, right? I mean, good. We know now creatine transporter directly matters for the anti-tumor CD8 T cells. We nail it down to the antigen response of the CD8 T cells. We nail it down to the specific aspect of the T cell activation. But how, right? How creatine, really link to all of those biological function of CD8 T cells. Here is what we did. So here is this, creating working network within a cell again, right? Creatine coming and can convert between the phosphocreatine versus creatine catalyzed by the creatine kinase. And we at first confirmed all the molecules matters to this network indeed exist in the CD8 T cells. We can detect the creatine transporter, upregulation in the T cells after stimulation. We can detect the upregulation of the creatine kinase, in the cells. And we did look into the two enzymes responsible for the biosynthesis of the creatine. T cells are not traditionally known to produce creatine, and we here confirmed that after the antigen stimulation, they didn't gain additional ability to make creatine, which means that they depend on the extracellular, supply of the creatine, which can only get inside of the cell through the creatine transporter. That explains why creatine transporter matters for them, right? And indeed, it is the energy side matters for this entire creatine transporter, story, because that we find that's a significant drop of the intracellular ATP level in the cell deficient of the creatine transporter. And when we give back ATP, it can kind of rescue the function of the T cells. So here is in molecular and signaling, level, like how exactly it happened. Okay, so here is a creatine transporter on the surface of a T cell. Here is a T cell receptor C tumor, for instance, tumor antigen that will induce signaling, cascade. Okay, this signaling cascade will induce all the effect functions of a T cell, including proliferation, including making useful cytokines, including the making of the useful cytotoxic molecules to fight off a tumor. Okay, and what creatine transporter does, it based on our study, is that it transport extracellular creatine into creatine. Remember, we talked about T cells have limited function ability to make their own creatine, because they really depend on the transportation of the extracellular creatine. Okay, the creatine get in, and remember, we also mentioned after activation, T cell also upregulate their creatine, kinase, right? So that can help the conversion between the creatine and the phosphocreatine, and a proper conversion of those will have a buffering level of the ATP in the cells to make the cell well supplied to do their function, right? And it can directly, ATP, of course, contribute to this entire signaling pathway by provide high energy phosphate for the tyrosine kinase, serine kinase, like necessary for activated downstream of TCR. And on top of that, ATP has another function. It can block this important signaling mechanism called AMPK. AMPK, once it's present over there, activated, it block the T cell function. So ATP, kind of being over there, it's regulated through the AMPK2. So combination of directly work as an energy molecule and also influence the intracellular signaling pathway of T cells all together, combined together, make creating an important molecule to keep the T cell at a normal and active functional status. We talked about all of those, and that led to a very interesting possibility of helping T cells to fight a tumor, right? T cells count on creatine transporter to transport extracellular creatine that can help them to, become more stimulated, more active, resistant to exhaustion, and just do a better job, for fight off the tumor. And how about we can supplement creatine, to the immune system, the T cell can uptake it through creatine transporter and then utilize creatine, to build up this energy buffering, situation inside of T cells to sustain their function. So here is what we did to test this possibility. So here we have 3 groups of animals, and it's either without creatine supplementation or with creatine supplementation through IP injection, which in animal, is mimicking the IV delivery, of the creatine situation. Or we can supplement creatine through the food supplementation. It's formulated into the diet of the animal. Okay, so we are very excited to find that creatine supplementation, either through IP injection or through the food, supplementation, and can significantly help the mice to suppress the tumor growth. And when we look into the tumor environment, we find that there's a significant increase of the CD8 T cell activity, and the supplementation of the creatine also benefit the mice, by increase the muscle, growth and the length, which interesting because for the late stage cancer patient, many of them may suffer from cachexia, loss of the body weight, loss of the muscle, mass, and this seems can do both, can help the immune cell to fight, also can help muscle to restore, their growth. And one more experiment showing over here is to just confirm indeed, those anti-tumor, benefit we saw is through the immune, system. When we try to supplement creatine to immunodeficient animal, we don't see the benefit. Okay, and when we, supplement animal to the supplement creatine to the wild type animal, but deplete the CD8 T cells, we see that the benefit is gone. So like it's this immune, protection mechanism absolutely, kind of worked through the CD8 T cell, the T cell anti-tumor immunity arm. One more interesting situation, we tested is how about the situation of the immune checkpoint therapy, right? So immune checkpoint blockade therapy, like anti-PD-1 treatment, anti-CTLA-4 treatment, and then there are more, in the pipeline, to be proved nowadays. But like anti-PD-1, for instance, really kind of revolutionized the treatment of many types of solid tumor already. That's a good news, but here is a daunting news. Not every, type of tumor, respond well to the current, immune checkpoint blockade therapy, like anti-PD-1 treatment. And within even a responding type of the cancer type, okay, not every cancer patient respond well. And for every cancer patient respond at the beginning, almost like majority of them relapse at the end. So there's a very, a big need for finding new ways to continue, improve the existing immune checkpoint, therapy, right? So we asked whether we can supplement the animal with creatine now, help the T cell in another way, right? So immune checkpoint helps them to get less exhausted, and the creatine can help them to get more energy, kind of to sustain better. We think that the combination of those may give additional, we call it synergistic benefits. And indeed, that's the case. So like we tried the different combinations, the animals, gave the anti-PD-1 alone, creatine alone, or the combination. And we clearly see a combination really gives the best therapeutic effects. So taking together all of the, above studies, we ended up with this hybrid engine model to explain how, creatine, play a role in the CD8 T cell mediated anti-tumor immune response. We consider here is a tumor microenvironment, okay? Here is a tumor cells, and here is a T cell trying to fight off those tumor, okay? And we kind of mimic the T cells as like hybrid car, right? It has a hybrid engine. The T cell need to take energy to fight off tumor, and the energy they can take is a nutrient, the biofuel resource, glucose, amino acid, lipid, okay, those can get into the cell, like become the fuel, make the cell to work. But is that enough? We think it's not enough. What creatine does over here, we think is work like more like a molecular battery, in contrast to the molecular fuel engine, okay? So that utilize the other resource. Creatine come in and working like a buffering system, right? So like when there's a lot of ATP inside of the T cells, the additional, energy can install in creatine in the form of the phosphocreatine, right? And when those fuel is low, which is very likely, right? Because the competing with tumor, for those nutrients in the environment, it's quite a hard competition, okay? And so those biofuels can be low all the time, okay? And that situation, then the molecular battery, which means creatine, now as a high energy form, can release the ATP into the system to keep the T cell fighting going on, okay? And of course, like thinking about supplementing creatine, it can come from the food, right? It can also come from the chemical version of the supplement. And for the possible clinical interventions in the future, we would like to, envision it maybe, supplied, to the cancer patient as oral supplement, or IV injection, or, even in a very well-designed combination therapy, like it can formulate it together with PD-1, anti-PD-1 blockade or other immune checkpoint, blockade therapy, and together to work, in a synergistic way to help the patient find the disease. Is that T cell the end of the story? Obviously, no, right? So we performed this large profiling study, in order to gain a more complete view of this immune landscape, like how the, various type of immune cells may, or may not be impacted by the creatine supplementation. So here is the experimental design. So we take an animal model. Again, we treat the animal, with creatine or not with creatine, and with or without, the combination with the immune checkpoint blockade therapy, like anti-PD-1 treatment. And we collect the tumor, from those animals, from the various treatment group, and we isolate the single cells from those tumor, and we have a special focus on the immune cells infiltrating into the tumor. And we performed the single cell RNA-seq that allow us to look into every single cell and every single subtype of the immune cells by grouping and the computational analysis. And here shows the real data, like what we got, okay? And as you can see, we can easily detect over, like 12 subtype of the immune cells over there, including our favorite CD8 T cell. But beyond that, right, there are other type of T cells over there. There's myeloid cell, and then that's, dendritic cells, which are a very important population of antigen presenting cell. Pick up tumor antigen, present to T cell, really essential for their stimulation, okay? And what I can tell you is that we detect multiple changes from various subtypes, and our next immediate, kind of target is dendritic cells, okay? Those dendritic cell populations, as you can see over here, if we focus on those dendritic cells and compare like the with or without creatine, treatment, we can see some quite dramatic changes for the dendritic cell function. And we can see the upregulation of the genes in the pathways representing the inflammation, immune, response, and then also, impact, from the metabolism side, which is no surprise, because if we temper the, ATP system, inside of cell, we do expect that's a metabolic change, right? So we are following all of those clues right now. And for people who are interested to know some signature genes, here is a plot showing you that for the innate immune function, particular dendritic cell function, glycolysis, ATP synthesis, that's quite large set of gene that dramatic changes, which we are currently following to understand what creatine really do, to that particular type, of the function of dendritic cells and how they work together with anti-PD-1, to make bigger change, and what's a possible synergistic effect that would become. I will stop the talk over here, saying that it's just the beginning. Creatine is obviously very helpful, to the anti-tumor, immune response in our preclinical animal models, and it's certainly expand beyond the T cells. Now we are starting dendritic cells, that's a couple of other type of cells we are also pursuing too. I would like to thank my research lab has done, the work like I just shared with you, and in particular, a talented PhD student, James, and our senior, scientist, Bo. So they are leading the dendritic cell work I just shared with you. And there are many undergrads and the alumni from the lab contribute to that. I would like to thank my collaborators and funding agencies. And of course, I would like to thank you in the audience for your attention. I will be happy to answer questions you may have. I pasted over here my email and my lab's website, and look forward to more communications with you. Thank you. Bye.